Welcome back to the deep dive. Today we're taking on, well, a pretty foundational topic. Animal reproduction, straight from a major biology textbook.
Right, and our mission here is to make sense of it all. We're talking evolutionary strategies, intricate anatomy, and the incredibly precise hormonal control that makes it all happen.
Exactly. So if you've ever felt a bit lost looking at a diagram of the menstrual cycle or a list of hormones, this is for you. We're going to build it up logically.
We'll start with the basic modes, how animals reproduce in the first place. Then we'll get into the logistics of fertilization.
And from there, a deep dive into human systems, how gametes are made, and finally, the hormonal orchestra that conducts everything.
It's all about two things, really: creating diversity and ensuring survival. Whether an animal is, splitting in two or carrying a fetus for months, the mechanics have to be perfect.
Okay, let's jump in with that first big division, sexual versus asexual reproduction.
Sexual reproduction is the one we're all familiar with. It's the fusion of two haploid cells, sperm and egg, the gametes, to form a diploid zygote.
Mixing genes.
It's all about mixing genes. Asexual reproduction, on the other hand, is where you get new individuals without any fusion. It's all based on mitotic division.
So, essentially cloning.
Pretty much. And you only find it in invertebrates, but the variations are really amazing.
So what are some of those?
Well, there's budding, for one. A new animal literally grows right off the parent, kind of like an outgrowth. You see this in stony corals.
Okay.
Then you've got fission, which is a more direct split. One animal just divides into two equal halves.
And then there's a really sci-fi sounding one.
Right. Fragmentation and regeneration. This is where an animal can break into pieces, and each piece, it just regrows into a whole new animal. Some worms are masters of this.
But the one that always gets me is parthenogenesis.
Ah, yes, the virgin birth. It's where an egg develops into a complete animal all on its own.
No sperm involved at all.
None. The classic example is the male honeybee, the drone. He's a haploid, born from an unfertilized egg.
But it happens in vertebrates too, right? I remember reading about Komodo dragons.
It does. Female Komodo dragons in captivity, with no males around, have been seen reproducing this way. It's a powerful backup plan.
Now, even within sexual reproduction, there's some fascinating gray area, like hermaphroditism.
Yes, where one individual has both male and female reproductive systems. For an animal that can't move, like a barnacle, this is a huge advantage.
Because any other barnacle it meets is a potential mate.
Exactly. You can donate sperm, you can receive sperm, it instantly doubles your chances.
And then there's actual sex reversal. The bluehead wrasse is the poster child for this, isn't it?
It is. They live in harems with one big male. If that male dies,
The largest female takes over.
she doesn't just take over, she physically transforms into a functional male in about a week. It shows you just how fluid these roles can be in nature.
Which brings up this huge evolutionary question, why even bother with sex? It seems so costly.
It's called the twofold cost of sex. Think about it. If you're an asexual female, 100% of your offspring can also reproduce.
They're all daughters.
Right. But a sexual female, half her offspring are males who can't produce their own young. You're losing half your reproductive output every generation, in a way.
So why pay that price? What's the benefit?
The best hypothesis we have is that the genetic variation it creates is worth the cost, especially in a changing environment. It's sometimes called the Red Queen hypothesis.
You have to keep running just to stay in the same place.
Exactly. If pathogens are constantly evolving to attack you, sexual reproduction shuffles the deck and creates new gene combinations, some of which might be resistant. It's about long-term survival of the species.
Okay, that makes sense. So let's get into the logistics. How do the gametes actually meet? We've got external versus internal fertilization.
External is the aquatic strategy. You release your eggs and sperm into the water and hope for the best. Fish, amphibians, they rely on this.
And it requires a wet environment, obviously.
Absolutely. The gametes can't dry out, and the sperm need water to swim. Timing is also everything.
You're talking about spawning. That mass release of gametes?
Right. The polola worm is a perfect example. Once a year, tied to the lunar cycle, millions of them release their tail segments packed with gametes. The ocean literally turns white.
An incredible level of coordination.
It has to be. Internal fertilization, on the other hand, was the key adaptation for life on land. The sperm is deposited directly inside or near the female tract.
Protected from drying out.
And this requires more complex behaviors and often chemical signals, things like pheromones.
So a female moth can attract a male from, what, a kilometer away?
Sometimes, yes.
Right.
Just with a scent. The trade-off is clear. With internal fertilization, you produce far fewer gametes, but the survival rate of each zygote is much, much higher.
Because they're protected either in a shelled egg or inside the mother's body.
Precisely. And this all relies on specialized organs, the gonads, to produce the gametes. But some of the accessory structures are just as interesting.
Like the spermatheca in insects.
Yes, it's a sac in the female, where she can store live sperm, sometimes for months or even years.
So she has control over when fertilization happens.
Complete control. She can wait for the perfect environmental conditions to lay her eggs and then release the sperm to fertilize them.
Which leads to that fruit fly experiment. It challenged the whole idea that it's just about the last male's sperm winning.
It did. The old idea was sperm displacement. The second male's sperm just physically pushes the first male's out of the way.
But that's not what they found.
Not at all. They found that even when a female remated with a sterile male, she somehow actively ejected or destroyed sperm from the first mating.
So the female reproductive tract is not a passive container.
Far from it. The female is playing an active chemical role in choosing which sperm succeeds. It's a major level of control.
Fascinating. Okay, let's pivot to the human system. And maybe we can frame this as a kind of production line, starting with the male anatomy.
Good analogy. The factory floor would be the testes, which contain the seminiferous tubules. That's where sperm are actually made.
And a key design feature is temperature control. They're in the scrotum for a reason.
They have to be. About 2 degrees Celsius cooler than core body temperature. It's essential for proper sperm development.
So once they're made, they're not ready to go yet, right?
No, they need to mature. They move into the epididymis, which is this incredibly long, coiled duct. It's 6 meters long.
6 meters.
Yep, and it takes them about 3 weeks to travel through it. That's where they gain motility and finish developing.
Then from there, it's the vas deferens and out through the urethra.
Right. And it's important to remember, the urethra in males does double duty for both the reproductive and excretory systems.
And along the way, they pick up fluids, creating semen.
Yes, from 3 accessory glands. The seminal vesicles add most of the volume, including fructose for energy. The prostate gland adds enzymes.
And the little ones, the bulbourethral glands.
Right. They secrete a clear mucus before ejaculation. The main job is to neutralize any acidic urine in the urethra. But, and this is key, that fluid can contain live sperm.
Which is why the withdrawal method of contraception is so unreliable.
Exactly. Now, for the female system, the gonads are the 2 ovaries. They contain all the follicles, and each follicle holds a single developing oocyte, or egg cell.
And when that egg is released, it's caught by the oviduct.
Correct, or the fallopian tube. It has this ciliated, funnel-like opening that sweeps the egg in. Fertilization almost always happens in the oviduct.
The egg then travels to the uterus.
The womb, yes. A thick, muscular organ lined with the endometrium, which is rich in blood vessels to nourish an embryo. The base of the uterus is the cervix, which opens into the vagina.
Okay, so we have the structures, but the real difference is in how the gametes themselves are made. Spermatogenesis versus oogenesis.
And they could not be more different. Spermatogenesis is, well, it's a continuous, efficient factory. It starts at puberty, and it just doesn't stop.
And one precursor cell makes 4 functional sperm.
4 motile sperm in about 7 weeks. Oogenesis is the complete opposite. It's cyclical, and it's interrupted.
Interrupted how?
It actually begins when the female is an embryo. All her primary oocytes form, and then they just stop. They arrest in the middle of meiosis, in prophysole.
And they wait there for years. Decades.
Potentially for decades, yes. Then at puberty, one resumes development each month. But unlike sperm production, you only get 1 mature egg.
What happens to the other cells?
The cell divisions are totally unequal. Almost all the cytoplasm goes to the 1 egg cell. The others become these tiny, non-functional polar bodies that just degenerate.
And even then, the process isn't finished.
That's right. The final stage of meiosis, meiosis II, only completes if a sperm actually penetrates the oocyte. The whole system is incredibly regulated, and it's finite, ending at menopause.
That long pause, that decades-long arrest, that's linked to an increased risk of genetic issues with age, isn't it?
It is. The cellular machinery has been on hold for a very long time, increasing the chance for errors when it finally restarts.
So let's talk about the conductor of this whole orchestra,
Yeah.
the hormones. It's a 3-tier system, right?
A classic hierarchy. At the very top, you have the hypothalamus in the brain. It secretes a hormone called GnRH.
Gonadotropin-releasing hormone.
Right. And GnRH's only job is to talk to the next level down, the anterior pituitary gland.
Which then releases the 2 big players, FSH and LH.
follicle-stimulating hormone and luteinizing hormone. These are the messengers, the gonadotropins, that travel through the blood to the gonads themselves and tell them what to do.
And the gonads then produce the sex hormones like testosterone and estrogen.
Exactly. And in males, the system is pretty straightforward. It's a stable negative feedback loop.
So it regulates itself to stay constant.
Perfectly. The FSH helps sperm mature. LH tells the Leydig cells to make testosterone. The testosterone then feeds back to the brain and says, okay, we have enough, you can slow down. It keeps levels steady.
But the female system is anything but steady. It's this dynamic cycle.
A drama in two acts, really. The ovarian cycle for the egg and the uterine cycle for the womb, and they are perfectly synchronized.
So let's walk through. It starts with the follicular phase.
Right, days 0 to 14. FSH, the follicle-stimulating hormone, does its job. It stimulates a follicle to grow.
And that growing follicle starts making estradiol, a type of estrogen.
It does. And at first, these low levels of estradiol have a negative feedback effect. They actually inhibit the pituitary.
But then something flips.
This is the most crucial counterintuitive part of the whole cycle. As the follicle gets really mature, the estradiol level gets very high. And that high level flips a switch in the brain from negative to positive feedback.
So high estrogen now tells the brain to release more hormones.
Exactly. It causes a massive surge of GnRH, which in turn causes a huge spike in LH and FSH.
And that LH surge is the trigger.
That is the trigger for ovulation on day 14. The follicle bursts and the egg is released.
So what happens to the follicle that's left behind?
After ovulation, we enter the luteal phase. LH transforms the ruptured follicle into a structure called the corpus luteum.
Which becomes its own little hormone factory.
A very important one. It pumps out high levels of progesterone and estradiol. And this combination puts the brakes on hard.
Strong negative feedback again.
Very strong. It shuts down the hypothalamus and pituitary to make sure no new follicles start developing while this egg is potentially on its way to be fertilized.
And all this lines up with what's happening in the uterus.
Perfectly. The rising estrogen in the first half causes the endometrium to thicken. That's the proliferative phase. Then the progesterone from the corpus luteum makes that lining ready for an embryo, the secretory phase.
But if no embryo implants?
Then the corpus luteum disintegrates after about 10 to 12 days. The hormone factory shuts down. Progesterone and estradiol levels plummet.
And without those hormones, the uterine lining can't be maintained.
The arteries constrict, the lining breaks down, and it's shed. That's menstruation. And the drop in hormones releases the negative feedback, allowing FSH to rise and start the whole cycle over again.
We should just mention, this shedding of the lining, the menstrual cycle, is pretty unique.
It is. Humans and some other primates do it. Most other mammals have estrous cycles where the lining is just reabsorbed if pregnancy doesn't occur, and they're only sexually receptive during a specific period of heat.
Okay, so let's follow this to its conclusion. Conception and, well, control.
So, if conception does happen in the oviduct, the embryo starts dividing and travels down to the uterus. Around day 7, as a blastocyst, it implants into that rich endometrial lining.
And its first job is to send a signal, right?
A critical signal. The embryo starts secreting a hormone called HCG, human chorionic gonadotropin.
What does HCG do?
It mimics LH. Its job is to keep the corpus luteum alive and producing progesterone. Without HCG, the corpus luteum would die, progesterone would drop, and the pregnancy would end.
So that's what a home pregnancy test is detecting.
That's the molecule. It's the embryo shouting, "I'm here, don't shed the lining."
And that embryo, its outer layer, helps form the placenta.
A remarkable life support system. It allows for the exchange of nutrients, gas, and waste between mother and fetus without their blood supplies ever mixing.
And that first trimester is so critical. That's organogenesis.
The period where all the major organs and body structures are laid down. By 8 weeks, it's no longer an embryo, it's officially a fetus.
And what about childbirth itself?
That's driven by another powerful positive feedback loop. Fetal signals start gentle uterine contractions. The stretching of the cervix triggers the mother's pituitary to release oxytocin.
And oxytocin makes the contractions stronger.
Which causes more stretching, which releases more oxytocin. The loop continues and intensifies until the baby is delivered.
Given all this complexity, it makes sense that we've developed ways to control it. Let's run through contraception based on where it intervenes.
Okay. First, you can prevent gamete release. That's how hormonal birth control works. The synthetic estrogen and progestin use negative feedback to prevent that LH surge.
No LH surge, no ovulation.
Simple as that. Or you can prevent fertilization with a physical barrier. Condoms, diaphragms.
And condoms have the crucial added benefit of preventing STIs.
The only method that is highly effective at doing so, yes. Then you have permanent methods, sterilization.
Vasectomy in males, tubal ligation in females.
Right. They just block the path of the gametes.
Hmm.
They don't interfere with hormones or function at all,
And finally, you can prevent implantation.
which is how a device like an IUD works. And of course, for infertility, we have technologies like IVF, in vitro fertilization, which bypass many of these early steps.
So to wrap this all up, the scope of animal reproduction is just immense, from a worm regenerating itself to this incredibly complex, clockwork-like hormonal cycle in humans.
But the theme is always coordination. Whether it's behavioral, like the Palolo worms responding to the moon, or the chemical symphony of the menstrual cycle, it's all about precise timing.
And the contrast between making gametes is just stunning. A continuous factory for billions of sperm versus this decades-long start-and-stop process to produce a single mature egg.
It really is profound.
So as a final thought for you to take away from all this, think back to that bluehead wrasse, the fish that can change sex.
A fascinating case.
We said the whole point of sex is to create genetic variability for long-term species survival. But the ability to change sex seems designed to maximize an individual's reproductive success right now.
So the question is, how does a strategy that maximizes short-term individual success ultimately help or maybe even hinder the long-term genetic fitness of the entire species?
It's that classic evolutionary tension between what's good for the individual versus what's good for the group. Something to think about.
A great puzzle. Well, thank you for joining us on this deep dive.
Go forth and explore the mechanics of life further.